set of digests where you took your mouse genomic DNA and you cut with a variety of restriction enzymes. And I'll show you what we can learn about genomic DNA from this. So your first lane is the 100 base pair ladder, 100, 200, 300, 400, 500 for Darwin, that's 1,000, 1,200, and 1,500. The first lane of your digest, lane 3, is the ALU digest. And ALU is a 4-cutter. The recognition sequence is given on the previous page, AGCT, so a 4-cutter, and the frequency with which a 4-cutter should cut genomic DNA like human would be quarter times a quarter times a quarter times a quarter, right? So the frequency of that site, you'd have the possibility of an A at any given position be a quarter, the probability that the next nucleotide is G is a quarter, the probability the next nucleotide is C is a quarter, and the probability the next nucleotide is T is a quarter. So that would be a quarter to the 4th, which is 256 base pairs. Of course, the sites aren't going to be spaced every 256 base pairs. You're going to get a Poisson distribution of sizes. And you can see here that the smear we get from ALU1 is a fairly low molecular weight. So you can see that there's material down here at 200 and then all the way up to, about 1,200. So a lot of material spread out over a very long range. So you'd expect, because it's a 4-cutter, that ALU would cut the most sites in the mouse genome compared to our 6-cutter and our 8-cutter, and you can see that that's definitely true. Then the next site is cutting the PICO-R1, which is GAATTC. That's a 6-cutter, so that would be 1/4 to the 6th, which is 1/4096. So you'd expect this to cut about every 4,000 base pairs. And now you're seeing a smear still, but a much higher molecular weight smear, because this enzyme cuts so much less frequently. Now you see a band here. There's an interesting story behind this picture. The reason you're seeing a smear is you're cutting the genome into so many different sized fragments, and the genome is so big that you can't resolve all those different sized fragments, right? So instead of seeing individual bands, you see a big smear, because you can't see thousands and thousands of bands, right? But we can see that band. And why can we see that band? What do you think? Anybody have an idea? Why? A repeat, yeah. So not a VNPR, but another kind of repeat. The repeat we're seeing here is a repeat that's found in a mammalian genome called line1, or long interspersed repeat 1, I think. Anyway, line1. It's a repeat that is, as I recall, about 2,000 nucleotides long. It's scattered all over the mammalian genome. And the reason we're seeing a band here at about 1,300 or so is because this repeat sequence has 2 EcoR1 sites in it. And so all those repeats get cut by EcoR1, and so you get a lot of these EcoR1 fragments that are exactly the same size from this repeat that's scattered all over the genome. And so you actually see a band in the eco lane, and that's from that line1 repeat. Well, how can I be so sure? We actually used to work on this line1 repeat in class, so I know for sure that's what it is. We used to do southern blots where you would run a digest on a gel and then blot transfer it to a membrane like you did with your protein, except we did it with DNA. And then instead of hitting it with monoclonal antibodies, hybridize with the probe. And if we hybridize with the probe to line 1, this band would light up like a Christmas tree. So, and then we cut out that band and sequence it and so on. But that's definitely the line 1 repeat, and it's in about 250,000 copies in the human genome. So it's one of those repeats that accounts for why our genomes are about 50% repetitive DNA. Okay, so that's the EcoR1 digest. Then the next digest is that SFI1 digest. SFI1 is an 8 cutter, and so it cuts 1/4 to the 8th power, which is once every 64,000 base pairs. So you'd expect your SFI1 digest to look a lot like uncut DNA, which is over here, and it does. You see maybe a little bit of digestion from regions of the genome where just by chance you have 2 SFI1 sites fairly close together. But you're not going to get much cutting, and remember, really big fragments you can't separate on these agarose gels, so it's not surprising that it looks like this. So this is really a good demonstration that if you want small fragments for your experiment, you can use a 4 cutter. If you want sort of medium to large fragments, you could use a 6 cutter, and if you want really, really big fragments, you can use an 8 cutter. Any questions about those 3? Okay, the next 2 lanes are going to tell us about methylation in adult mouse liver tissue. So this digest is the HEPA2 digest, and this is the MSP1 digest, and they both recognize CCGG. But obviously this one's cutting more than this one. And remember I said there's a difference in terms of how they cut relative to methylation. So which one of these do you think is the one that's sensitive to methylation and won't cut? This one or this one? Yeah, this one, the HEPA2. So HEPA2 doesn't care, or HEPA2 rather does care, because if the site is methylated, the CCGG, if it's methylated at that second C, because remember methylations happen at the CG dinucleotide. So if that CCGG is methylated at that second C, then HEPA2 will not cut. And so you can see that the cutting with HEPA2 actually looks a lot like the cut with SFI1. On the other hand, MSP1 doesn't care if it's methylated, and it cuts more. You can see that there's definitely a bigger smear here from the MSP1. So this tells us then that a lot of the genome in adult tissue, adult liver tissue in this case, is methylated. And we know that from other experiments now. This was actually the first way that people looked at mammalian genomes and said, wow, there must be a lot of methylation. Now we have methods that we're talking about PacBio, right, where you can actually look at the methylation state of the genome in a sequencing reaction. And now what we find is that in adult tissue, there's a lot of methylation because all those genes that aren't needed in that tissue type are heavily methylated and shut down. There are other repetitive regions of the genome that are methylated and don't get transcribed. So this kind of experiment was really the first to demonstrate heavy methylation of genomes. Now, if you looked at early cells, if you looked at zygotes that had just gone through joining of the egg and sperm and maybe gone through a few cell divisions, if you look at those, you see not as much methylation, not much at all, because in those cells, they're still totipotent, the genes are open for expression, you see much less methylation. So if we were looking at those kinds of cells, we wouldn't see such a big difference there.
Methylation can happen everywhere, or is it specific site?
Yeah, methylation can happen at CG dinucleotide. So if you have a sequence that's A, T, C, G, C, the CG can be methylated at the C residue. So in mammals, it always happens at the CG dinucleotide. In E. coli, it happens at A's, the gamma methylation. So it's different in different kinds of organisms. But mammals methylated at CG dinucleotide. So a lot of methylation. The next, sorry. Oh, sorry. Always behind my computer.
Can you quantify the methylation, or just say that?
No, this would just say, yeah, there's a lot of methylation. If you want to quantify the methylation, then you've got to do something a lot more complicated. Can't be able to tell you about that. Okay. And if you wanna know methylation of specific genes, again, this PacBio system is probably the best way to do it.
I actually learned there's a four-part that looks closest to people related to the six-part, or it's you have a high-part cut before using that.
Yeah, MSP, always looks like it's not cutting up to its potential, and I don't think, it's the enzyme. It's known that in the human genome, there are fewer CCGG sequences than you'd expect, so it's really not, it doesn't occur at once every 256. It's much less frequent than that, and that's partly because the human genome isn't 50% GC and 50% AT, it's actually 60% AT. So if you wanted to calculate the frequency of CCGG in a 40% GC genome, you're really multiplying instead of a quarter by a quarter by a quarter by a quarter, 0.2 times 0.2 times 0.2, and so it's less than that. And it actually ends up, I think, in the human genome being about once every 1,000 base pairs. Okay? Any other questions? Okay, so then we've got this lambda-hindi3 marker, we've looked at it before, and then this is the uncut DNA, clearly not digested. There's a little smear here. I don't think that's from degradation. I think it's just because the gel is overloaded with a lot of DNA. And when you can't run in a nice tight band, it just sort of buzzes out. So if the gel's overloaded, you'll get things that look like that. Okay, any questions from this gel? Okay, so that was one result I wanted to show you. The other thing I'm going to show you before I let Andy take over is I'm going to show you an adenine 2 sequence from class. Round of taking pink colonies and doing PCR, we got 3 successful CRISPRs that we saw, and 2 were from Nicole. And so I've got one of Nicole's sequences here. Her sequence worked too. The adenine 2 sequences look a little better than our GAPDHs, but still not great. We've been working with Ricardo on this and trying to figure out what's going on. Still not clear. So we swapped out all those reagents. That didn't do it. We still have kind of messy sequence here, and I'll show you what I mean in a second. So it doesn't look like it's the sequencing reagents. We also have shown that it's not the instrument because we ran some other sequences on the instrument and they look fine. So it's not the instrument. It doesn't look like it's the sequencing reagents. That doesn't leave us much to blame, and kind of what we're left with are those columns. So remember I told you that we had the columns that we bought for the course, and then we switched to another batch of columns. Doesn't seem to, I mean, it helped a little bit, but we're still having some issues, and I'll show you what the issues are by highlighting things on the screen here. So what we're looking at here is the sequence again of one of Nicole's 2 clones, so we would expect that this would have the insert. And you can see down here, this is 230, 240 nucleotides, and this looks pretty good. This is really good sequence. Now you can see some background underneath it. You see that? That's like a secondary sequence underneath our actual sequence. And I don't know what this is, but it looks like contamination. It looks like there's another sequence in there that's also being sequenced at the same time. That'll be more obvious when I go over to the left here more where it gets worse. You can see back here now that underlying sequence is giving us real problems, right? So if you look at this position right here, is that a T, which is the red peak, or is that a C, which is the blue peak that's right in the same place? So if you're sequencing 2 things simultaneously, that's what you get. You get at at some positions, the nucleotides are gonna be different between the two sequences, and so you're gonna see something like this, right? Now here, where this T is, you don't see that, and that's probably because the real sequence that we want, and whatever this other thing is, both have a T at that position, so we get this big T and no underlying sequence there. But then the nucleotide before it, we've got a red T and again, blue C. So it's really problematic at the beginning of the sequence, but as we go further in, it becomes less and less of a problem. Now our sequence is actually pretty good out here. And then further on, as the peaks get lower, because we're getting further out in the sequence, as we get further out, now this underlying sequence again starts to be problematic. So it's possible. I don't know. Now it's a weird problem that we've not experienced often. Poor Ricardo is tearing his hair out. I think there's a possibility that the columns are contaminated with something, or the column buffer that they give us to walk through the column. If there's some contaminating DNA in there, then that could be causing us a problem, but it's a puzzle. Yeah, right.
Can you find out what the contamination is by just like manually looking at the smaller entities?
Yeah, we could go through and we could try to pick out that sequence, and I would lose my mind after a while. I've already half lost my mind from trying to read these. You can give it a shot, Bryce. If you want to try to read that, this would be, good, extra credit, right, for the final exam. You could read that extra sequence underneath there and then blast it and tell me what it is. That would be really useful. I'm not sure you want to do that. Bryce is like, I'm doing it. Totally doing it. Bryce will be here till 11 o'clock tonight. We'll be down there having a good time. He'll be up here on the Anyway, so let's go, so what I did is I took this sequence and I thought, well, maybe we'll get lucky and maybe I'll be able to read the region clearly where the BAM site is. Because remember, when we successfully CRISPR, we took out some sequence and replaced it with our sequence that was on the editing fragment. And on that editing fragment, we had the BAM site, which is GGATCC, which is not in this region of the yeast genome. And also we inserted a stop codon. So I just want to show you quickly what's on the board under here. Small section of chromosome 15 in yeast. Chromosome 15 is where the adenine 2 gene resides. So this purple represents the adenine 2 gene. And this is the 20 nucleotide region that we replaced. And it's got a BAM site in the middle of it. And then right after the BAM site, there's a stop codon. I think it was TAA. Yeah. Oh, that's it, yeah, TAA. So there was a G, right, a G, and then TAA. So it's a G, and then TAA after the BAM site. And then there's 20 nucleotides code here. We did PCR with a forward primer that's a few hundred nucleotides down this way, and a reverse primer that's a few hundred nucleotides this direction, to get a PCR product that was 688 base pairs, I believe. That should have been the total size of our PCR product. And those of you who got a PCR product, it was in that size range, and more of you will be able to see it hopefully on this next cell, since we redid PCR. But about 688 base pairs. I'll look up the exact number, but that's what's popping up in my head. And then we took that 688 base pair PCR product. Good thing we have in because my voice is not getting any better. When we cut with the PCR product with BAMH1, we're going to get a product we saw this on Nicole's sample, yesterday. We cut with BAM-H1, we get one fragment that's 240 base pairs roughly, and another one that's 450 base pairs apart. And from our 3 pink colonies that we saw yesterday, we saw in fact that we did get those 2 BAMs when we cut the PCR product, and hopefully we'll see more of that again today, because you redid your PCR, and then we took the PCR product and just cut it with BAM, and now the staff's running that gel, and we'll see those results later on. Obviously we're not gonna have a chance to resequence those, and it's probably just as well, because we haven't solved the problem, and so we could resequence them and probably end up in the same situation until we figure this out. It's really embarrassing. The high school group that's here did their sequences and theirs worked.
Oh.
I mean, it's a reagent issue. They're using different reagents, different columns. They're sequencing reagents with a PCR product. We expect the BAM site in there. And so let's take a look at this sequencing in whole again. Here's, I've typed GGATCC into the search box because that's a BAM site. Remember that stretch of the each chromosome has no BAM site in it. So if we get a BAM site here, it's because we put it in, and we believe we did because Nicole's digest showed the 2 BAMs. But let's see if we can verify that. So there it is. And we got lucky on this because you can clearly see, you all see that, the peak? The sequence is G, which is black, G, A, which is green, T, C, C. Okay, so that sequence is pretty clear, even though we've got this underlying mess below it, right? Now the next nucleotide should be a G, right? It should be GGATCCG. Well, the instrument couldn't call it. The instrument called it a K because it's not sure if it's a G, which is this black T, or a T, which is this red T. If it's not sure if it's a G or T, it puts K. Well, we can look at this and say, well, the G peak is higher, so we can call that a G, and in fact, we know that's what it should be. So let's call it a G. Then the next 3 nucleotides ought to be TAA. Well, there's T, and now we have that same situation with the double peak, but the A is a little higher, right? And then the next one is clearly an A. So GTAA, and that's our stop codon. Now let's keep reading. This is supposed to be a G, right? Then a T. You watch up there. I'm watching down here. G, C, and then I'm going to call that CA, and then TT. So, I mean, we're clearly in the right spot. So this clearly is the sequence of our replacement editing fragment. If we go the other direction, we can see the same thing. So if we go backwards, we should have, let's just read backwards. We'll go C, C, A, G, T, etc. So if I read it going backwards and you look up there, you can tell me if I'm right. So C, C, A, G, T, C, T, and then we get this K, which to me looks like another T, and then C, A, G. So we're in the right spot. Now the reason I say we got lucky is the reason we found this spot is because I typed in GGATCC, and it found it because this happened to be a sequence that's clear. The nucleotide right after it didn't call. So if this messiness had been shifted over a little bit, we wouldn't have been able to find this just by searching. So we got a little lucky, but I've examined this whole sequence, and you can read the whole sequence of what was inserted. And then on the other side, you go back into the each chromosome, and on the other side, you go back into the each chromosome.
Just wondering, can this happen when you instead of 1 colon put 2? You put them in 1, 2?
It could, but think about this. If we pick 2 colonies, let's say one had the insert and one didn't, right? They all have the plasmid because they all came up resistant. They've all got plasmid, and so yeah. So the ones that got the plasmid and successfully did the CRISPR will have the same sequence through for the first 300 base pairs as if they weren't CRISPR'd, right? The non-CRISPR'd section, let me put up we're going to read we do PCR and get the product, and then we're going to read that product by hybridizing the forward primer. That's how we did the sequencing. And so when you start the sequencing, you're sequencing through these chromosomes. And so whether you have successful or CRISPR or not, this part of the sequence would be the same, right? So if you pick a non-CRISPR'd sequence and a CRISPR'd sequence, these first 300 some odd base pairs should have been the same, and that's where our sequence looks worse, actually. So I don't think that's it. I don't think it's because you all picked 2 colonies accidentally, which would be weird. But it's a good idea, but I don't think that's it for that reason. That makes sense to everybody. Yeah, right.
Use that to the 40 base pair line. That's the lower implication and
And that could be because reading that lower sequence is really difficult. So, yeah. But you can keep trying. You gotta come up with an answer for it. It would be interesting to find out what that is, but that's gonna be hard, because reading that sequence, I mean, I'm having a hard time reading the good part of the sequence, right? So, anyway, so, let me back up a little bit on the CRISPR. So I told you earlier today that if you don't have a nice color thing like we did, and for most genes, 99.9% of genes, you're not gonna have some kind of nice color change like we have with this experiment. I specifically designed this experiment this way because of that color change, right? So that we could pick pink colonies and stack the deck in our favor. But if we were CRISPRing any other gene in yeast, we wouldn't have that. And so how would we know if we got CRISPR? Well, what we do is take our colonies, and we would have to pick colonies at random and do PCR, and that's essentially what you just did upstairs, right, or downstairs. You pick some colonies at random, CD, and now we're PCR. So that's how you have to do it.
Yeah, so practically, like, how many colonies you would pick randomly, but you know that, like, good or not?
Right, so it depends, depends on how efficient your CRISPR is, right? And so, different CRISPR systems, like whether you're using Cas9, Cas11, or whatever, will have different efficiencies. It's also different depending on your cell type, the organism you're working with. So, in yeast, it might be more or less efficient than, say, mouse cells, CHO cells, let's say. So, you know, it's hard to say. You'll know your efficiency if you pick 100 colonies and you get 5 that are CRISPR, then you know your efficiency is maybe around 5%, right? What we find in our yeast experiments when we've done this in the past is that we get about 10%. So we expect about 1 out of every 10. So if each group picked 2, that's another 32 that we picked. So you know we might get another 3 or 4 that way. See how it comes out. And I know that picking that many colonies or that many cell lines and sequencing them sounds like a lot of work, and it is, but it's a lot of work, but you have a high payoff because then you've got what you want, you've got that cell line now, you can keep it forever, right? So okay, any questions about that? I probably better stop and Andy go. And when I come back later, hopefully my voice will be